Good evening. Thanks for coming. My name is Kyle Durgin, and I had the privilege of participating in a Dimensions Fellow in Research in the University of Iowa Neuroscience Department. Just a little background of where I was working. So I was working in the Neuroscience Department, and within the department, I was in Dr. Robert A. Cornell's lab, which is in the Anatomy and Cellular Biology Department, which is located in the Bowen Science Building, pictured here. The scope of my project, I was conducting research in a professional lab environment, and my task was to develop a method and study a specific question, and then use the scientific method to change my methods, modify my methods to prove or disprove a hypothesis. And I was specifically working with the TRPM1 ion channel in zebrafish melanophore regeneration. And we were specifically interested in this TRPM1, which stands for transient receptor potential melastatin-like 1 ion channel, which the reason we were interested in that is that TRPM1 is has significantly just decreased expression in malignant melanoma cells, and melanoma is a really nasty form of skin cancer. So we were trying to figure out what its role is, and then hopefully with an increased knowledge, then we can maybe come up with a method to cure or slow down the cancer. And the model system that we work with is zebrafish. Here's a zebrafish pictured here. And the reason we like to work with zebrafish is that, first of all, they're translucent, which makes it much easier to study what's going on inside them. They also have rapid observable development, which is very important in research. And then they have a fully sequenced gene, like the sequences of all of the major genes, and that's very useful for us. And what's really fascinating about zebrafish, and I experienced this firsthand, is that you can watch their development happen very rapidly. Pictured here is the zygote, and then the first cell stage, and then down at the bottom is an embryo about 3 days post-fertilization. And I'm going to be talking about microinjection, and we microinject around the 1 cell stage, between the 1 and 2 or 4 cell stage. And so what I did specifically is I injected zebrafish embryos with a 25 base pair nucleotide fragment, which is called, we call it a morpholino, which is a way of knocking down the gene. And the morpholino binds to the RNA, and it prevents it from encoding a protein. So by injecting the morpholino, we hope to knock down the RNA and shut off the TRPM1, or prevent the formation of the TRPM1 ion channel. And the reason we do this is that the best way, or one of the best ways to understand what a protein or a gene does, is to knock it out and see what happens when it's not there, which seems pretty obvious, I guess. And then a second part of my project was working on cloning the TRPM1 gene, so I could overexpress the gene, which is the opposite of knocking it down. Create more RNA and inject that into the embryos and see what happens in that situation. Here's my lab team. Only Amanda Decker, who's back there, made it tonight. It's a long drive from Iowa City, so it's okay. And then here's Jen. She's actually a Cornell alumni, which was cool. It's a lot of Cornells, so it was a little confusing. Cornell lab, Cornell alum, and I'm from Cornell. So, academically, I learned how to microinject, which at first I thought it was pretty easy. You have a needle and you poke it into the fish, and then you pump a pedal and it squirts the morpholino in. But it's harder than it sounds. A lot of the embryos were dying. I felt bad, but I got over it. I also learned a lot about the TRP channels, TRPM1 specifically, but there's TRPM1 is part of there's 8 other ion channels within the TRPM family. I learned data analysis with the help of Amanda and critical thinking skills. And this was a great research experience because it took what I learned at Cornell or here in my classes in developmental biology with Craig Tepper, and then I used all the techniques and stuff in the lab setting. And it related research and genetics and that stuff to the medical field. I've always been interested in going to medical school, and this was a good connection between research and the medical field, just because of the TRPM1's decreased expression in melanoma cells. And I've actually been down there the last block, and I'm there again this block, continuing my research. And I'm hoping to potentially write an honors thesis if I can get enough good data. Professionally, I made some good connections in the science community, which I hope will strengthen my resume and give me an advantage into getting into med school, which is where I ultimately want to go. It helped me focus my career goals in medicine. I was kind of wondering if I really wanted to research, and as much as I enjoyed it, I'm still tending to go towards medical school. It was great to interact with other people in the same field. You could sit down in the lunchroom and people would talk about their research, and it was great. And then I had to present every week, what I had learned in lab meeting, which was a good experience as well. And then I also had to solve problems. A lot of times my experiments wouldn't go as planned, or I'd run a gel and I wouldn't know what I was looking at, and I'd have to figure out what was going on and see what I could do to correct it. I'd like to give special thanks to the Dimensions program for funding my internship, and my site supervisor, Robert Cornell, and my faculty sponsor, Craig Tepper. And then my lab team, Christine, Greg, Tiffany, Jerry, Jen, and Amanda, who is actually my spiritual and menu advisor as well. And I'd also like to thank RJ and the rest of the Career Engagement Center for all their help too. And that is it.